Welcome back to the Deep Dive. Today, we're diving deep, really deep, into this invisible world all around us and inside us. Microorganisms, our health, disease.
That's right. We're looking at how our bodies and these tiny microbes interact, what disease actually is, how it spreads.
And the science that tracks it all, epidemiology. Basically getting you up to speed on the unseen forces that shape, well, your health.
Exactly. And we're not just sticking to definitions. We want to connect the dots,
Yeah.
Link these foundational ideas to what happens in the real world, drawing from great sources like microbiology and an introduction.
So you're going to discover some pretty surprising things today.
Definitely. About the battles, but also the alliances happening right inside you.
Okay, let's unpack this first bit. Our bodies are constantly negotiating, I guess, with microbes. We've got our defenses.
Sophisticated defenses.
And then you have these disease-causing ones, the pathogens. It's this ongoing thing.
It really is. And when our defenses win, we stay healthy. But if the pathogens overwhelm us, that's when disease happens.
Right.
And that whole dynamic, that's what the science of pathology studies. Pathology looks at the etiology, the cause of a disease.
Okay.
Its pathogenesis, which is basically how it develops, and the changes it causes in the body, structurally, functionally.
So it's not just about having a bug, is it? There's a difference between infection and actual disease.
A crucial difference, yeah. Infection is just the invasion. The pathogen gets in, maybe starts colonizing. But disease, that's when the infection actually leads to a change from your normal state of health. Something detectable.
Got an example?
Sure. Someone could be infected with HIV, the virus, but show no symptoms of the disease AIDS for years. Or think about E. coli. It's normally in your gut, totally fine.
Yeah, part of the normal crew.
Exactly. But if that same E. coli gets into your urinary tract, then it can cause a nasty infection, a full-blown disease.
Okay, that makes sense. And here's where it gets really mind-blowing for me. When we talk about your body, it's not just your cells we're talking about.
Not even close. It's astonishing. For every one of your, say, 30 trillion human cells, you're hosting an estimated 40 trillion bacterial cells.
Wow. Seriously. More bacteria than human cells.
Yeah. This huge, bustling community living on you and in you, that's your personal microbiome.
A world within us. So where does it all come from?
Well, it actually starts setting up shop even before you're born. There's evidence now for a placental microbiome, but the really big seeding event is birth itself.
Great.
Babies born vaginally get a big dose of microbes like lactobacillus and bacteroids from their mothers. C-section babies, though, their first microbes tend to look more like skin microbes, Staphylococcus aureus, for instance.
And that matters, that early difference.
It seems to. Research, like from the Human Microbiome Project, suggests these early microbial communities might be linked to risks for things later in life, like type 1 diabetes, asthma, maybe even obesity.
That's incredible. They've even tried swabbing C-section babies to sort of catch them up.
They have, yeah. Trying to see if they can help their microbiomes look more like those of vaginally born infants. It's a really active area of research.
So we've got these microbes that are always there, the normal microbiota. Are permanent residents, usually harmless.
Right. Under normal conditions, they don't cause trouble.
But then there are others that just visit, transient microbiota.
Exactly. They're just passing through, present for maybe days, weeks, or months, but they don't set up permanent residence.
What decides who stays and who goes, and where they live?
Oh, it's all about the environment. Different parts of your body are like different neighborhoods. They offer different things. Nutrient availability is key.
Makes sense. Food source.
Physical factors, too.
Yeah.
Temperature, pH level, oxygen availability. Think about your skin. It's relatively dry, acidic, salty sometimes. Tough place for many microbes.
Right.
But your large intestine, it's warm, moist, anaerobic, packed with nutrients. It's like a microbe metropolis down there.
Huh. A metropolis. I like that.
And mechanical forces matter too. Chewing, saliva flow, urine flushing things out, the cilia in your respiratory tract sweeping things away. It all influences who can stick around.
So our normal microbes are actually really important then, not just passengers.
Incredibly important. They did these studies with germ-free animals raised in completely sterile environments. And while they can survive, their immune systems are really underdeveloped. They're super susceptible to infections if they do get exposed. It really highlights how much we rely on our normal microbial partners.
Okay, so this relationship we have,
Yeah.
it's a type of symbiosis, right? Where at least one partner depends on the other.
Precisely. Symbiosis is that close relationship, and it comes in different flavors.
Flavors, okay.
Yeah, first you've got commensalism. One organism benefits, the other is pretty much unaffected, like Staphylococcus epidermidis on your skin. It eats your skin secretions, but doesn't really help or harm you.
Okay, that's one.
Then there's mutualism. This is win-win. Both partners benefit. The classic example is E. coli in your large intestine.
The one that can cause trouble elsewhere.
The very same. But in the gut, it's a good citizen. It makes vitamin K for us, some B vitamins too, things we need. And in return, we give it a nice warm place to live and plenty of nutrients.
Mutualism.
Right.
Got it. And the third?
That's parasitism. This is the one we usually think of with disease. One organism, the parasite, benefits at the expense of the other, the host. Many disease-causing bacteria fall into this category.
It really sounds like a delicate ecosystem.
Yeah.
You mentioned balance. What happens if it gets thrown off? How do our good microbes actually protect us?
That's a key concept. Microbial antagonism. Or you might hear it called competitive exclusion. Basically, your normal microbiota actively fight off potentially harmful invaders.
How do they do that?
Several ways. They compete for nutrients, they produce substances harmful to other microbes, and they can affect the environment, like the pH. For example, in the vagina, normal lactobacillus bacteria produce acid.
Right.
That acidic environment keeps yeast like Candida albicans under control. But if you take antibiotics that kill off the lactobacillus,
Wow.
the pH changes, becomes less acidic, and Candida can overgrow. Result, a yeast infection.
So antibiotics can disrupt this balance.
Big time. Another example. E. coli in the gut produces substances called bacteriocins. These inhibit the growth of closely related bacteria, including nasty pathogens like Salmonella and Shigella.
Wow, they police their own neighborhood.
Kind of, yeah. And this is super relevant clinically. Think about Clostridium difficile or C. diff.
Heard of that. Causes terrible diarrhea, right?
Yeah.
Especially in hospitals.
Exactly. C. diff is often present in small numbers in the gut, kept in check by the rest of the microbiota, but broad-spectrum antibiotics can wipe out its competitors.
Leaving the field open for C. diff.
Precisely. It overgrows, produces toxins, causes severe colitis.
Yeah.
And that's why one of the really effective treatments now for recurrent C. diff is actually, well, giving patients pills containing healthy gut microbes. Fecal microbiota transplants, essentially. Restoring the balance.
That's amazing. Restoring the ecosystem. So some microbes are usually fine, but they can become troublemakers if the conditions are right, like microbial sleeper agents.
That's a good way to put it. They're called opportunistic pathogens, normally harmless in their usual spot, but if they get a chance and opportunity, they cause disease.
Like E. coli in the urinary tract again.
Perfect example. Or think about someone whose immune system is suppressed, maybe due to AIDS or chemotherapy. Microbes that wouldn't bother a healthy person, like pneumocystis, can cause life-threatening pneumonia in them.
So opportunity knocks, and they take it.
Basically, yes. And sometimes microbes even cooperate to cause disease. Like in periodontal disease, some pathogens need to latch onto other bacteria, like oral streptococci, just to get a foothold on your teeth.
It's complex. So with all these potential culprits, how did scientists figure out which microbe causes which disease? It feels like finding a needle in a haystack.
It was, until Robert Koch came along in the late 1800s, a German physician, a real pioneer. He established a systematic way to prove that a specific microbe causes a specific disease.
Okay, how?
He worked on anthrax, showing Bacillus anthracis was the cause. And tuberculosis, linking it to Mycobacterium tuberculosis, he developed what we now call Koch's postulates. It's like a checklist.
A checklist for guilt?
Sort of, 4 steps. 1, the same pathogen has to be present in every single case of the disease.
Okay, consistency.
2, you have to isolate that pathogen from the diseased host and grow it in a pure culture in the lab.
Get it by itself.
3, that pathogen from the pure culture must cause the same disease when you introduce it into a healthy, susceptible laboratory animal.
Not this one.
And 4, you have to be able to re-isolate the same pathogen from that newly diseased animal. Prove it's the original organism.
That sounds pretty solid. Does it always work? Like, can you always apply those 4 steps?
Well, that's the catch. It's a powerful framework, but there are important exceptions. Some microbes just refuse to be grown in lab cultures on artificial media.
Like what?
The bacterium that causes syphilis, Treponema pallidum, or the one for leprosy. Many viruses are like this too. They need living cells. Sometimes researchers had to use animals or even chick embryos, like for Legionnaire's disease originally.
Okay, so culturing can be a roadblock. What else?
Well, some diseases can be caused by several different microbes. Think pneumonia or meningitis. Different pathogens can lead to similar symptoms.
Right, not always one cause, one disease.
And conversely, sometimes one pathogen can cause different diseases. Mycobacterium tuberculosis usually hits the lungs, but it can affect other organs too. Streptococcus pyogenes, strep throat, scarlet fever, skin infections, all from the same bug.
Complicates things.
And then there are ethical issues. Some pathogens, like HIV, only infect humans. We obviously can't intentionally infect people to satisfy Koch's third postulate. That's completely unethical, though historically things were different.
Yeah, thankfully things have changed. Okay, so when someone is sick, how does a doctor figure out what's wrong? They look for clues, right?
Exactly. They look for signs and symptoms. Symptoms are subjective changes. The patient feels pain, malaise, fatigue, things you can't directly measure.
Like feeling lousy.
Right. Signs, on the other hand, are objective changes the physician can observe and measure, like fever, swelling, lesions, paralysis.
The things you can see or quantify.
Yes, and when a specific set of signs and symptoms always accompanies a particular disease, we call that a syndrome.
Okay, now, thinking bigger picture. How do we talk about diseases not just in one person, but in a whole population?
We classify them based on how they behave in a population. First, how they spread. Communicable diseases can be transmitted from one host to another, directly or indirectly, like chickenpox or the flu.
Spreadable.
If they spread really easily, we call them contagious. Think measles. Then there are non-communicable diseases. These don't spread from host to host. Tetanus is a good example. You get it from a contaminated wound, not from another person.
Got it. And how do we track how common a disease is?
We use two key measures. Incidence tells you the number of new cases in a population during a specific time period. It shows how fast the disease is spreading.
So like the rate of new infections.
Exactly. Prevalence, on the other hand, is the total number of cases, both old and new, in a population at a specific time. It gives you a snapshot of how widespread the disease is overall, taking into account how long people stay sick.
Okay. So incidence is the speed, prevalence is the overall burden.
That's a good way to think about it. And prevalence can reveal a lot of hidden disease. If only 1 in 10 people with something like cryptosporidiosis actually seeks treatment, the reported incidence might look low, but the true prevalence, the actual number of people affected, could be much, much higher.
Wow. The iceberg effect.
Kind of, yeah. We also classify diseases by how often they pop up. Sporadic means they occur only occasionally, like typhoid fever in the U.S. these days.
Infrequent.
Endemic means a disease is constantly present in a population, like the common cold. It's always around at some level.
Always there.
Epidemic is when many people in a given area acquire a certain disease in a relatively short period. Think of a flu outbreak overwhelming a city.
A surge.
And if an epidemic spreads worldwide, that's a pandemic. Influenza has caused several pandemics, and AIDS is considered a pandemic too.
Global scale. Okay, one more vital concept here, especially with vaccines in the news. Herd immunity.
Yes, crucial concept. Herd immunity is when enough people in a population are immune to a disease, usually through vaccination, that it becomes difficult for the disease to spread.
So the immune people form a protective barrier.
Exactly. They protect the vulnerable individuals who aren't immune, maybe because they can't be vaccinated or their immune system is weak. It limits outbreaks. The eradication of smallpox is the ultimate success story of herd immunity.
A huge public health achievement. Now, what makes one person more likely to get sick than another? Are there factors that sort of tip the scales?
Absolutely. We call these predisposing factors. They make you more susceptible or can alter the course of a disease. It's a whole range of things.
Like what?
Gender can play a role. Females tend to have higher rates of UTIs, males higher rates of pneumonia. Genetics matter. The sickle cell trait, for example, offers some resistance to malaria.
Interesting.
Climate and weather respiratory diseases often increase in winter when people are indoors more. Age is a big one. The very young and the very old are often more vulnerable. Nutrition, stress levels, lifestyle choices, having other illnesses, even undergoing chemotherapy. It all contributes to your individual susceptibility.
A lot of variables. Okay, so let's say someone does get infected and the disease starts. Does it follow a typical pattern?
Generally, yes. Infectious diseases often progress through 5 stages. First is the incubation period.
The time between getting infected and feeling sick.
Exactly. It's the interval between the initial infection and the first appearance of any signs or symptoms. And this period can vary a lot depending on the disease. Importantly, you can often be contagious during incubation, even before you know you're sick.
Spreading it without realizing.
Okay,
What's next?
the prodromal period. This is usually short, right after incubation. You get early, mild symptoms, maybe general aches, feeling unwell. It's often hard to tell what you have at this stage.
I think I'm coming down with something phase.
Pretty much. Then comes the period of illness. This is when the disease is most severe. You have overt signs and symptoms, the ones characteristic of the disease. Your immune system is fighting hard. This is often when you're most contagious, too.
Peak sickness.
Right. If your immune system wins or treatment works, you move into the period of decline. Signs and symptoms start to subside. You feel better, but you might be weak and vulnerable to secondary infections.
Getting better, but not out of the woods.
Exactly. And finally, the period of convalescence. You're recovering, regaining strength, returning to your pre-disease state. But like incubation, you can sometimes still be contagious during convalescence. Some people become long-term carriers, like with typhoid fever or cholera.
Still potentially spreading it even when feeling better. Good to know. Okay, so for diseases to keep going, they need a place to hang out between hosts, right? A source.
Yes, that's the reservoir of infection. It's the continual source of the disease organism, where it lives, multiplies, and has the opportunity to be transmitted.
And what kinds of reservoirs are there?
We group them into three main types. First, human reservoirs. This includes people who are actively sick, but crucially, it also includes carriers.
People who have the pathogen but aren't sick.
Correct. They harbor the pathogen and can transmit it, but show no signs or symptoms themselves. Typhoid Mary is the classic historical example, or people carrying whooping cough without symptoms.
Silent spreaders. Okay, what else?
Animal reservoirs. Diseases that primarily occur in animals but can be transmitted to humans are called zoonoses. Think rabies in bats, skunks, raccoons, or Lyme disease, where field mice are a key reservoir for the bacteria, and ticks transmit it. There are about 150 known zoonoses.
Wow, lots of animal connections. And the third type?
Non-living reservoirs. These are environmental sources. Soil can harbor fungi causing ringworm, or bacteria like Clostridium, which causes tetanus and botulism. Water contaminated with feces is a huge reservoir for diseases like cholera, typhoid fever, cryptosporidiosis, and improperly handled food, too.
Soil, water, food. Okay, so the pathogen is in the reservoir. How does it actually get to a new host? What are the travel routes?
There are three main routes of transmission. First is contact transmission.
Touching stuff.
Basically, it can be direct contact, person-to-person physical contact, touching, kissing, sexual intercourse, colds, flu, STIs spread this way, even an animal bite, like for rabies. There's also congenital transmission from mother to fetus.
Okay, direct.
Then there's indirect contact. This involves a non-living object called a fomite.
A fomite.
Yeah, things like contaminated tissues, towels, bedding, drinking cups, toys, even stethoscopes or money. Or contaminated needles for things like hepatitis B or HIV.
So touching contaminated objects.
Right. And the third type of contact is droplet transmission. Microbes spread in mucus droplets from coughing, sneezing, laughing, talking. These droplets travel short distances, usually less than a meter. Influenza, pneumonia, whooping cough often spread this way.
Okay, so contact.
Yeah.
Direct, indirect via fomites and droplets. What's the second main route?
Vehicle transmission. This is transmission via a medium like water, food, or air.
A vehicle carries it.
Yep. Waterborne transmission involves pathogens spread through contaminated water cholera, shigellosis. Foodborne transmission is through food that's uncooked, poorly refrigerated, or prepared unsanitarily. Think food poisoning from salmonella. Cross-contamination is a big issue here, often involving the fecal-oral route.
And air.
Airborne transmission. This is different from droplets. Here, pathogens are carried on droplet nuclei or dust that travel more than 1 meter from the source. Measles virus and the tuberculosis bacterium can spread this way. Fungal spores, too.
Further distance through the air. Got it.
Yeah.
And the third route?
Vectors. These are animals that carry pathogens from one host to another. Mostly arthropods, insects like mosquitoes and flies, or ticks and fleas.
Bugs as taxis.
Pretty much. There are two ways they do it. Mechanical transmission is passive, like housefly landing on feces, picking up pathogens on its feet, and then landing on your food. The fly is just a carrier.
Gross. Okay, and the other way?
Biological transmission. This is more complex, an active process. The pathogen reproduces inside the vector. Then, when the vector bites a host, it transmits the pathogen. Malaria is a classic example. The parasite develops in the Anopheles mosquito, which then transmits it when it bites someone. Lyme disease via ticks is another.
So the vector is part of the pathogen's life cycle.
Exactly.
Okay, this brings up a really important modern issue. infections people get while they're in a hospital or a healthcare setting. Healthcare-associated infections, HAIs.
Yes, a huge challenge. They used to be called nosocomial infections. The CDC figures are pretty sobering. Maybe 1 in 25 hospital patients gets at least one HAI. We're talking millions of infections, tens of thousands of deaths each year in the U.S. alone. It's a leading cause of death.
Why are hospitals such hotspots?
It's really a perfect storm involving three factors. First, you have microorganisms in the hospital environment. Hospitals concentrate pathogens, including opportunistic ones that might be part of normal microbiota but cause trouble in the sick. And increasingly, antibiotic-resistant strains thrive there.
Like MRSA.
MRSA is a big one, yes.
Yeah.
And Pseudomonas. And Clostridium difficile is actually the single most common cause of HAIs now.
Wow. Okay, so bugs in the hospital, what's factor 2?
The compromised host. Hospital patients are often already sick. Their immune systems might be weakened by disease or treatments like chemotherapy. They might have breaks in their skin from surgery or burns or invasive devices like catheters or ventilators that bypass natural defenses. They're just more vulnerable.
Makes sense. And the third factor?
The chain of transmission within the healthcare setting. This can be direct contact from hospital staff to patient or patient to patient, or indirect contact via fomates catheters, respiratory equipment, even bed rails.
So what's being done? How do we fight back against HAIs?
Prevention is key, built around what are called universal precautions. This starts with standard precautions, basic hygiene measures applied to every patient, every time.
Like what?
The absolute #1 thing is meticulous hand hygiene. Washing hands or using alcohol-based rubs before and after every patient contact. Compliance isn't always perfect, but it's the single most important step. Also using appropriate personal protective equipment, gloves, gowns, masks, respiratory hygiene, safe injection practices, proper disinfection of
The
equipment.
basics done right every time.
Exactly. Then, for known or suspected infections that are highly transmissible, there are additional transmission-based precautions. These are layered on top of standard precautions. There are contact precautions for things spread easily by touch, like C. diff or MRSA. Gloves and gowns are crucial. Droplet precautions for things like flu or bacterial pneumonia requires wearing a mask. And airborne precautions for measles or TB, which need special ventilation rooms and N95 respirators.
Tailoring the protection to the threat.
Precisely. Plus other things like disinfecting tubs, cleaning respiratory equipment really well, using disposable items when possible, and having dedicated infection control staff monitoring trends and intervening quickly.
It really highlights how preventing spread, like with Jamil Carter's C. diff case we mentioned, relies on these consistent practices: gloves, thermometers, careful antibiotic use.
Absolutely. It all ties together to protect patients.
Now, beyond HAIs, we keep hearing about new diseases popping up or old ones changing, emerging infectious diseases, EIDs.
That's right. EIDs are diseases that are new or increasing in incidence or threatening to increase in the near future. It's a dynamic landscape. And interestingly, about 3/4 of them are zoonotic, originating in animals. Many are viral and often spread by vectors.
What causes a disease to emerge? It seems like there are lots of reasons.
There really are. It's complex and often involves multiple factors. You can have genetic changes in microbes creating new strains. Think of new flu variants or virulent E. coli like O157 by H7.
Solution in action.
Yep. The widespread use of antibiotics and pesticides drives resistance in microbes and the vectors that carry them. Climate change is a big one, altering temperatures, and rainfall can change the geographic range of vectors like mosquitoes, potentially spreading diseases like malaria or dengue to new areas.
Wow.
Modern travel and trade mean a disease that emerges in one part of the world can be on another continent within hours or days. Think Zika, West Nile virus.
Globalization of disease.
Exactly. Ecological changes, deforestation, natural disasters, changing agricultural practices can bring humans into contact with new microbes or reservoirs. Even things like breakdowns in public health measures like lapsed vaccination programs leading to diphtheria outbreaks can cause diseases to reemerge. And unfortunately, we also have to consider the potential for bioterrorism.
It's a lot of interconnected factors. Who keeps track of all this?
It's a global effort. Organizations like the World Health Organization, WHO, and in the U.S., the Centers for Disease Control and Prevention, CDC, and the National Institutes of Health, NIH, are constantly monitoring, investigating outbreaks, doing research, and coordinating responses to EIBs worldwide. The CDC even publishes a monthly journal specifically on this topic.
Okay, that brings us nicely to the scientists who do this tracking and investigation. The disease detectives, epidemiologists.
Yes, epidemiology. It's the science that studies when and where diseases occur in populations and how they are transmitted. It's about understanding the patterns, the causes, and the control of health problems.
And it's not actually new science, is it? People were doing this detective work even before they knew about germs.
That's absolutely right. Modern epidemiology really got its start in the mid-1800s with some remarkable individuals. John Snow in London investigating cholera outbreaks.
A Broad Street pump guy.
That's him. By mapping where cases occurred, he deduced it was spreading through contaminated water, specifically from one public pump, long before the cholera bacterium was identified.
Brilliant detection.
Then there was Ignaz Semmelweis in Vienna. He noticed huge differences in death rates from childbirth fever between hospital wards. He hypothesized it was being carried on the hands of medical students coming from autopsies. So he instituted mandatory hand washing with chlorinated lime, and the death rate plummeted. Again, this was before the germ theory was widely accepted, just based on observation and intervention.
Incredible. And Florence Nightingale.
Yes. Not just a nursing icon, but a brilliant statistician. During the Crimean War, she meticulously collected data showing that far more soldiers were dying from infectious diseases like typhus and cholera due to poor sanitation than from battlefield wounds. Her powerful visual displays of data spurred massive reforms in military hygiene and hospitals.
So they were lowering disease rates just by understanding the patterns, even without knowing the specific microbes.
Exactly. Their work laid the foundation. Today, epidemiologists determine disease etiology, identify risk factors, age, sex, occupation, genetics, lifestyle. They figure out how it's transmitted, where it's happening, look for seasonal patterns.
All to figure out how to control it.
Right. Control strategies range from developing drugs and vaccines to controlling reservoirs like mosquito control, improving sanitation and water treatment, ensuring food safety, promoting better nutrition, encouraging changes in behavior, the whole public health toolkit.
How do they actually conduct these investigations? Are there different approaches?
Yes, 3 main types. Descriptive epidemiology involves collecting and analyzing data about who got sick, where, and when. It's often retrospective, looking back at data, like Snow did with cholera maps, trying to describe the occurrence.
Painting a picture of the outbreak.
Right. Analytical epidemiology goes a step further. It analyzes a particular disease to determine its probable cause. This often uses comparison groups. A case control study compares people who have the disease with similar people who don't, looking for differences in past exposures. A cohort study follows groups of people, some exposed to a potential risk factor and some not, over time to see who develops the disease. Nightingale's work comparing sanitation levels and death rates was essentially analytical.
Looking for the why. And the third type.
Experimental epidemiology. This involves testing a hypothesis, often through a controlled experiment. Semmelweis's hand-washing intervention was a form of experimental epidemiology. Today, this often means clinical trials, where you test a new drug or vaccine. You have a test group getting the intervention and a control group getting a placebo or standard treatment, ideally in a blinded way so bias is minimized.
Rigorous testing.
Yeah.
Okay, so doctors and labs collect all this data on diseases. How does it get funneled into action?
Through case reporting, healthcare providers are legally required to report occurrences of certain notifiable infectious diseases to local, state, and national public health agencies.
So they have to report things like measles or tuberculosis?
Yes, exactly. There's a list of diseases that must be reported. This system provides the raw data for monitoring disease trends, detecting outbreaks early, spotting emerging infections, and guiding public health interventions.
And the CDC is central to this in the U.S.
It is. The CDC collects and analyzes this data from across the country. They publish the Morbidity and Mortality Weekly Report, the MMWR, which is basically the go-to source for current data on disease morbidity, the incidence of specific diseases, and mortality, the number of deaths from those diseases. This is how we track everything from the flu season to outbreaks of foodborne illness to trends in antibiotic resistance like MRSA in hospitals. It links directly back to those epidemiological principles.
Wow. It really connects everything from the tiniest microbe on your skin to how it might cause disease, how that disease spreads through a population, and how these dedicated scientists track it all down to protect public health. We've covered a huge amount today.
We really have. It's such a dynamic field, constantly evolving as we learn more about microbes and our relationship with them.
What's fascinating here, I think, is just realizing how much is constantly going on that we can't see, and how much more there still is to learn about these incredibly complex interactions.
Absolutely. Understanding this microbial world empowers you, really. It helps make sense of health news, understand prevention measures, and appreciate the invisible forces shaping our lives. And it definitely keeps raising important questions for the future.
Well, a massive thank you for joining us on the Deep Dive today. And thanks especially to you, our listeners, for being part of our Deep Dive family. We hope you feel a bit more informed, maybe a lot more curious. We'll catch you next time.